who did not do quiz one. So I announced that, but I will say it again. You don't have to do makeup quiz. And I don't even know whether it will be possible to do makeup quiz. And I do have, I guess I should also announce this because there were a few people who had issues with scantrons, and I was asked to mark their exam instead of scantrons. And I did ask associate chair, is that allowed? And here is what was how it was communicated to me. If you mark exam for one student, there is no reason not to do the same thing for everyone else. Then I'm not fair. But what I can, so I can't do that. So whatever you put on Scantron, that's what it is. Now, one thing that I could do, and that's what I was doing last year, if it happens in past, that if it happens in future that you are on the border for some mark, for example, you are at the like 75, 76, and you need 77 to get B plus, and doing that quiz better can help, I am willing to reconsider. So I can do that. That's allowed when somebody is on the border, and that helps. But in other cases, it's not fair, because in that case, if I take like a Brittany's exam and I mark her exam, I ignore her scantrons, there is no reason why Mariam wouldn't come to me and say, okay, I also did better on the quiz or like on the paper, so why I cannot be and then everyone will do that, and then I am in trouble. Then scantrons make no sense. So it just makes no sense. But again, if you are on the border, I'm very willing. And somebody asked me you asked me a few minutes ago, if you are at 86%, like let's say you have 86.5, is that 87? Yes. So if you have 86.5, I will give you 87. If you have 86.2, I will give you 86.2. Like, I'm not going to round it up to lower, because those 0.2 might help you for some other mark. So I will do everything what I can. I'm always going to be fair. There is no issue with that. Like my goal is for you all to do well. So that's what makes me look nice person and good prof. If I teach you well, that's what I do. That means that I'm good in my job. And that's what I want. Okay, so I'm going to be very fair. Don't worry about that. But I also want you to be fair to me, which means when you come to see and again, I'm telling you this because there was an example. When you come to see exams, quizzes, please don't fight with us. Don't fight for marks. And the reason is because when you start fighting for marks, first makes you look unprofessional. Second of all, it won't help you in later terms, because when I feel threatened, I will I will try to defend myself. And when students attack me, I have to defend myself. And then I'm kind of stepping back, and I develop like protective coat, like manganese oxidizing bacteria. So I can sink on the bottom of the ocean. And in that case, I am not going to, not that I don't want to help you, but then I cannot collaborate with you in future. That's the thing, because I feel threatened. So last year I had that issue when students was like, but I want this 1 point. I was like, but you didn't put it on Scantron, like I can't give you. And there was some issue with Scantron. You're like, yeah, but I put it here. I was like, well, but I don't actually know whether you put it here on the paper now when you were looking at your exam, because I couldn't see you. Whatever is on Scantron, I have to mark. And she was like, no, I want that point. I want that point. I was like, okay, I'm going to give you that point. But that's it. You're done. Give me my point. Here is your point. Long story short, at the end, student was on the edge for like literally, that's why I remember I 76, and I got an email. Can I get that 1 point? No. No, you can't. No, because you did not deserve that 1 point. I gave it you as a gift. And now you're asking me again. But you were very mean and told me bad stuff, and now you want me to help. So it's not like, But if we don't fight, then I'm here to help. But when somebody fights, I am scared. I am scared. I'm really getting scared, and I don't know how to behave, and that's not. So let's not fight. Let's work together. Like that's why I'm here, and I'm your friend. I'm not here to torture you. I think I'm very normal person. Okay, so let's talk about evolution of life at high temperatures and the hydrogen oxidation mechanism. So this is what we yes, sorry. Yeah. Like 69.5. Yeah, so we have to work with 41.7. 40, okay, I see. So see, I think that's good average. I mean, very happy that average, and thank God everyone else is happy. Because when I had last year 83%, there was a question, what did I do? Like, did I actually tell students what I left? Let's be honest, I do tell them sometimes what I left, right? So, if I tell you four reasons why I'm the symbiotic theory, okay, I give you slides, 1, 2, 3, 4, this is exactly what I asked. And I give you questions. So you, like, why don't you know? My point. And it will be the same for the preschool. Even for preschool, it will be easier because you will study from review slide. So, like, please study at my point. Like, I get very sad when students don't study. And here is why I'm telling you this. My husband told me a long time ago that he had a prof in high school who taught biology. He was molecular biologist. He still is. Uh, and my husband hated biology. Like for him, that's like ridiculous, uh, especially molecular biology. But he told me that he learned. So when we met, he knew about central dogma of molecular biology. I was like, how do you know that when you hate it? He was like, well, prof in high school was so nice, and he played basketball with us. He was like few years older than them, and I was so embarrassed not to study for his exams because he was so nice to us, and he tried to teach us, and we were just people who don't study. And I was like, okay, I'm embarrassed. I'm going to study now. And he learned about central dogma just because of that instructor. So, and I was young student at the time. I was thinking about it. I always knew I want to teach. That was my that's what I knew I will do that when I grow up. And I was thinking when he told me that, wow, so that means if you're a really normal person and if you respect your students, they will want to learn. Then why everyone doesn't behave like that? Maybe nobody told them, but I learned, and that's how I build myself as a instructor. So if I am normal, I want to motivate you to study. So please study. Is that okay? Okay, thank you. Temperatures and hydrogen oxidation. So very new research that I'm going to briefly, there are like only 20 slides, and then you can go work on your term projects, and you can cheer for us in Paris because we're traveling, but cheer more for me not to pass out. We are both guys this year, and yeah, it will be interesting. Uh-huh. Their feet dragged me into that, so she motivated me. Uh, So anyway, let's talk about how the life at high temperature developed. So definitely it was an ancient and long time ago because so a little bit of reminder. 4 billion years ago, last universal common ancestor appeared, Luca. So Luca must be thermophilic. Why? Because at the time Earth was very hot. So Luca had to be adapted or to say it correctly, had to evolve to live at high temperature. And next 100 million years, there was a high temperature. So Luca must be hyperthermophilic. No way it's different. Uh, we know for now that majority of organisms, microorganisms that are associated with high temperatures or hyperthermophiles are archaea. There are some bacteria, we talked about them last time, but archaea are the most famous by that. Again, there are many books here. We will cover most of them in this, like this is where we will end up. And again, this lecture, I think this lecture is the last one that goes on quiz two. Am I correct? It's not? Okay, this is okay. If it says 14, then this is the last one. So I will check in syllabus, but I will definitely announce it. Yes. Okay. So so here is the deal. We could do, I'm thinking that, and I will announce it so everyone knows. I think adding this one to the quiz too will facilitate your life for midterm. Because if you cover this is like only 20 slides, and in review session is going, like I will tell you exactly what to know. It will facilitate your life for midterm, so you don't have to study more. So I will add this lecture just to finish unit. And I thought this is lecture. I thought that I put this one because it's the end of the unit, so it makes sense to put it in the quiz. So it will be on exam, but I will announce so everyone knows. Bacteria and archaea live at high temperature. They live through certain adaptations. So they develop certain adaptations. So extreme temperatures are above 100 degrees. So and usually those microorganisms live in that 100, over 100 degrees geothermal water. And also there are some terrestrial hot springs that temperature is close to 100, not really 100, but in geothermal heated water, it's really superheat. So there are certain temperatures where you won't find microorganisms. Those are those superheated vent water, so that's completely sterile. So it can't be found there. So that brings the question, what is the upper temperature? Like when the enough is enough? Like what microorganisms actually cannot survive? So the one that, like, for now, again, what I know, this is the last what I know, which might be changed because new species are getting updated every day. Record holder is 122 degrees. It's the mechanopyrene. So it's for now that this was when I checked it last time. That one is able to survive like a substantial, like a higher temperature, like higher temperature, but 122 maybe plus minus 2. I found it's 122. So if you get a water that's 250 degrees, there are no biological life over there. So there is no DNA, there is no RNA, there is nothing. Why? Because ATP molecule that creates energy for life gets degraded at 150, so you can't really. So above 150 degrees, the life form that lives has to have like a lot of heat lability of molecules that are like that are distributed in cells. So it has to develop certain molecules that can survive. So many molecules are like are not stable at 150 degrees. So, so how would you survive at such a high temperature? Again, at 250, nothing can survive. DNA, RNA, nothing. So, but in order to survive even something above 122 or 100, you have to develop stable molecules of life like DNA and RNA, and of course proteins. So as you know, proteins start denaturing, some of them at 40 degrees. So in order to survive at 100 something, those have to be very thermostable proteins. Very often they develop formations called thermosomes or specific proteins that help in maintaining stability called chaperones or chaperonins. And they have specific versions of DNA gyrase, which is a specific enzyme that participates in DNA metabolism. And there are certain RNA modifications that help them to survive. What are thermosomes? So thermosomes are adapted. That's how microorganisms survive at higher temperatures. So what they do, they keep proteins properly folded in order to function at high temperatures. Thermosomes also can help in maintaining DNA. So shock treatment, when cells are returned to optimal temperature, the DNA that survived in thermosomes will be sufficient to enable cell growing and dividing. So that's the whole point of having these thermosomes. It's like protection of DNA and protection of proteins from degradation at high temperatures. Next adaptations are, of course, thermostable proteins. That means that proteins evolved evolved not to be denatured at high temperatures. So how they evolve? How they succeed to stay intact when the temperature goes like up to 100 degrees. So they have very specific folding. But what's interesting is that there are no any specific amino acids found. Like they're all still the same, like normal, like a regular proteins, but they have special folding. One of the key structures in proteins, and you probably heard that proteins can form alpha, like amino acids can be organized in alpha helix or beta sheet or loops, and beta sheet can be parallel, anti-parallel. So those are secondary structures. So one of the key secondary structure that was found in thermostable proteins is alpha helix. What you said is highly hydrophobic core, which means they cannot unfold, like unwind, and they have high ionic interactions on surface. So basically, everything that can prevent protein from like disintegrating, but there are no specific amino acids. Which proteins we are talking about, like which thermostable proteins are there are certain proteins that help in stabilization of other proteins at high temperatures. So those specific proteins are called chaperonin. So chaperonins are other name for them is heat shock protein. So what is their deal? So they are helping in folding. So if it happens that protein unfolding, even though it has very tight hydrophobic and ionic interactions, somehow it got unfolded, then chaperonins or heat shock proteins get synthesized to help refolding of these proteins. And chaperones don't only have the role in thermostabilization, there are different forms of chaperones. Many of them are produced in microbial organisms literally to protect proteins from other intracellular interactions, like very commonly that happens in protein translocation because you want protein to be exported in a proper state, and then you have a chaperone just shielding it and sending it through the membrane. But they can also function in thermal protection. Then DNA and RNA molecules that are like important for function of life, they can be stabilized through this, and this is very common in hyperthermophiles, reverse DNA gyrase. DNA gyrase is an enzyme that participates in DNA metabolism, like in stabilizing unwinding the helix. And there are certain other modifications that can help, like increased potassium molecule, the potassium ion, because with increased potassium ion, that can prevent DNA melting, and reverse DNA gyrase is actually stabilizing DNA molecule. That's the goal. What gyrase is doing is introducing so the normal DNA gyrase is winding DNA helix, and we call that in molecular biology like negative supercoiling. So gyrase is actually doing that negative supercoiling. This one is actually reverse, so it does positive supercoiling. And when DNA is positive, like when supercoiling is positive, that makes it more stable. And also, their hyperthermophile very often have highly basic DNA binding proteins. Where will you see highly basic DNA binding proteins? In eukaryotes, like histones, for example. Histones are highly basic proteins, which means they have a lot of basic amino acids like lysine and arginine. Why? What means basic? That means proteins that have a lot of plus charge on their backbones, because DNA is negatively charged, and then those positively charged proteins just bind it and cap DNA. So that's what histones do for us, and that's what highly basic DNA binding proteins do for hyperthermophiles, like helping protecting their DNA. And they also have these heat-resistant lipids, which we saw when we talked about ether bond in archaea, because archaea are mostly those that live in high temperatures. So you will see that those specific lipids, even in bacteria, sometimes if they are exposed to high temperature. Ribosomal RNAs, again, we heard about this. It's a structural and functional component of ribosomes. And so in hyperthermophils, in small subunit of ribosomes, you will see way more GC base pairs. Again, doesn't matter how many, like doesn't matter if it's 15 or 20%. What matters is that when you have a lot of GCs, because in the hydrogen bond that forms in between AT and GC in like in DNA, it's a double helix, but also in RNA, you have that intramolecular interactions between nucleotides. So A and T form two bonds, like there are two lines. GC form three, which means it's more stable, it's harder to break. That's why whenever you see whenever you see the gene that has a lot of GC content, you will have higher time to denature it, and then you also have to increase the time of denaturation in PCR. So again, just to know why, what if you don't remember from molecular biology. Now, let's go back to evolution. What was the hypothesis for the evolution of molecules at high temperature? So, biological molecules, biochemical processes in the first cell showed up on Earth around the thermometer. Like everything was hot and boiling, and that happened on the seafloor. So the theory is the modern hyperthermophils, because hyperthermophils, they're on the bottom of the tree. So the theory was that they're the closest descendants of the ancient cells, because they're at the bottom of the tree. So hyperthermophils live in specific habitat, and because they're so ancient, they must, like we know now that the first last universal common ancestor Luca was hyperthermophile. So they had to oxidize hydrogen. So usually when we talk about evolution at higher temperatures, like hyperthermophils, yes, they have all those modifications, but what in terms of metabolism and energy, they have to use hydrogen. They use hydrogen because why? Because it was available, hydrogen, and it was hot. Well, yes. So those that when they developed all those adaptations, they had to learn how to take electrons from only thing that was available, which was hydrogen. But in that case, they have to find something to give that electron to so they can form proton motive force. What was available was iron sulfur nitrate, rarely oxygen. Rarely, I say, because there are some connections later on. But mostly this was anaerobic respiration. So the ability of hydrogen oxidation is very tightly connected to hyperthermophils, and it's very highly spread among hyperthermophils. So definitely those hyperthermophils are the best representation of early Earth phenotype, and hydrogen must be playing a specific role because hydrogen is so reduced and was so abundant at the time that it was available and there was plenty of inorganic electron acceptors in that environment. So hydrogen is linked to the very early life development together with hyperthermophils. So like what Luca was, probably used hydrogen, probably was hyperthermophile, did anaerobic respiration. So all those organisms that we talk about when we talk about hydrogen oxidation and now hyperthermophils, they are very early developed, and hydrogen oxidation definitely must be involved in primary, like ancient, like a primary primitive organisms. So, how we call those organisms? We call them chemolithotrophs. And they live in very hot environment because they use hydrogen like inorganic compound and they respire something else. So chemoorganotrophy, which means oxidation of organic compounds, like involving organic compounds in respiration, it cannot go above 110. Like it doesn't really matter which number, but if we talk about evolution of life at high temperature, it's usually chemolithotroph, rarely chemoorganotroph. Who is the least tolerant on heat? Photosynthesis, definitely. If I ever ask these details, I will But what I wanted you to know is basically that the main form of nutrition for hyperthermophils is chemolithotrophy. That chemoorganotrophy can happen only up to some point, like roughly hundreds, and photosynthesis is definitely the most sensitive, like it's not tolerant at all, the most sensitive. So talking about hydrogen, hydrogen metabolizing bacteria. So what do they do? Do they live at high temperature? Yes. Lithotrophy? Lithotrophy. So it's chemolithotrophy. I don't think it can be chemoorganotrophy. I think most of organic compounds at 120 would just degrade. Unless it's some very thick stable protein that you find here, I think 100, maybe the literature say 110. But I think that everything above 100, like it's pure chemo. That's what, again, I would say 100, 100. Now, you remember how I told you how hydrogen is like all hyperthermophilic? Is it always? Doesn't have to be. Why? Because there are some organisms that are not hyperthermophilic, but they can still oxidize hydrogen. They're weird. Uh, So now moving back, so when we finished with so hydrogen is related definitely to hyperthermophils, but one new research that came recently was related to hydrogen metabolizing bacteria that, unlike those organisms that we just talked about that can oxidize hydrogen, respire iron or sulfur or nitrite, this one actually use oxygen as terminal electron acceptor. Seriously, yes. And they're interesting organisms, and they have a lot of applications in biotechnology. We'll see later why. This was published in 2023, and I talked about this first time last year. So they use, of course, hydrogen as a solar energy source. They use oxygen terminal electrons, so they're aerobic. As a carbon source, they will use technically carbon dioxide. They fix carbon dioxide, and they use carbon cycle because it's the most it's least efficient, as we know. The enzyme that all those bacteria has, and they're bacteria, they're not bacteria. It's hydrogenase enzyme. And 2 years ago in C1 conference, everything was about hydrogenase. Everything was related to hydrogenase. That enzyme has the ability to bind hydrogen. Uh, but hydrogenase is actually oxygen sensitive. You see the problem now? Because they need oxygen to respire, but this one is oxygen sensitive. So they actually need microaerophilic environment, which means they don't need a lot of oxygen. What else they have to have nickel because hydrogenase enzyme needs nickel metal as a cofactor. Some of them can fix nitrogen. Some, not all of them. So what do we know about that today? There are diverse aerobic, microaerophilic bacteria that can use atmospheric nitrogen as energy, atmospheric hydrogen as energy source. So why is hydrogen oxidation important? It's a globally important because it regulates composition of the atmosphere, of course, enhances soil biodiversity, and it drives primary production at extreme ecosystems because, again, hydrogen is very abundant, and whoever is on the bottom of the tree or on the bottom, like in the bottom of the soil, can use hydrogen because they have all those enzymes. So it literally drives primary production. So, but as I told you, there is a hydrogen in the atmosphere, but can bacteria use that hydrogen that exists around? They actually can't. It's really so teeny tiny amount. That's what people thought. Okay. So can atmospheric hydrogen be oxidized by microorganisms? People thought till last year, no, that's not going to happen. But what they found in that paper, which I found last year, and that's why I introduced it, there is an uncharacterized enzyme, hydrogenase, that has nickel and iron cofactors. And they exist in organisms that live under very huge metabolic challenges because what you have very low amount of hydrogen in the atmosphere. So that means the enzyme has to have very high affinity hydrogen. and you have oxygen in the environment. So in order to use that hydrogenase, you have to be able to detect that teeny tiny amount of hydrogen and use oxygen as terminal electron trap. So how do they do that? They actually oxidize teeny tiny amount of hydrogen and perform electron transport chain. So people in this paper, which I have to post if somebody wants to see, it's just from paper from Nature. It's really good paper, like amazing discovery. They have this nickel iron hydrogenase, which was a metal enzyme, has hydrogen. It was um, membrane bound, so it like metal enzyme, and so the one that was known previously was membrane bound, and it had very low affinity for hydrogen. So if something has very low affinity for hydrogen, that means that you have to have a lot of hydrogen to be able so the enzyme can detect it. And that previously known hydrogenase nickel iron was very poisoned with oxygen. Now, what they found in that paper is completely different version of that hydrogenase. It was also nickel iron, so also metal enzyme, and it had extremely high affinity for hydrogen, and it was able to extract electrons even from the like teeny tiny amounts of hydrogen present in the atmosphere, extract electrons and deliver to electron transport chain so bacterium could grow. Now, it was another interesting thing about the enzyme, not that it had high affinity for hydrogen, but it was insensitive to oxygen. So completely different from the previously known hydrogenase. And this is the paper that I will share with you. The organism is called Mycobacterium megmatis something, I have no idea. It's Mycobacterium. I couldn't pronounce that, but what I want you to know is that the adaptations of that enzyme that was found, that it was extremely high affinity for hydrogen and completely insensitive to oxygen inhibition. The complex, and it was like a complex, they call it complex, they call HAC enzyme. So that enzyme, HUC, HAC, it's enzyme that oxidizes hydrogen. It's a brown of color because it has multiple metal clusters, and it was very stable. It's a room temperature and went up to 78 degrees. So this is not hyperthermophilic enzyme. It can oxidize hydrogen, but it's not hyperthermophilic. So what is great is that it can actually detect any tiny amounts of the hydrogen present and it's sensitive to oxygen. So they did, of course, control with hydrogenase from E. coli, and of course, it was poisoned by oxygen. It was not able to detect it. And this is how the enzyme looks. So this is HAC enzyme. So that's how this is their schematic drawing, representation. It's not really pretty picture, but it tells the story. So it looks like a four-lace clover, and it has those like a salt-like protrusions. It's in cell membrane. It's like embedded. Activity, so it's associated with the membrane, but its activity is actually mainly associated with the soluble fraction of the atom. So unlike the previous one, which was extremely membranic, this one is kind of combination of soluble and membranic. So membranic. So they call this four-leaf clover oxygen tolerant hydrogenase. So again, amount of oxygen is much higher. Amount of oxygen in the atmosphere is much higher than hydrogen, but that HAC enzyme actually has those dimers. It forms like a dimeric structure that are sufficiently bound that they can displace, literally as they interact, they can displace all oxygen that is not reduced, and then they don't get poisoned. The other adaptation that was found is that that enzyme actually has something called gas channels, and they saw it. They had in the paper, they had a very nice microscope where they could see those HAC channels, and literally they track how hydrogen passes through that channel. So that's another difference comparing to previous enzyme. So they call that gas channel a road to the active site. So hydrogen literally slides through those gas channels. That's why they call it gas channels, because hydrogen is a gas. What they saw, again, even though when they did analysis compared like gas channels to see if normal hydrogenase has it, they saw that yes, it does have it, but in this new HAC, gas channels are much tighter. and it's much narrower in Hack than in other one. That was oxygen tolerant. So literally what they explained happens like as hydrogen enters in that narrow channel, oxygen gets displaced, and that's why it's excluded, like with the bottleneck. So that's how oxygen actually doesn't get to the active site. So pretty much it's all about that those gas channels that help in oxygen protection, because it's oxygen. That's how it gets oxygen insensitive. So that's the bottleneck. Like literally those gas channels that are much tighter, but they had very nice microscope that help them to track it down. We unfortunately don't have that here, and that would be all. So again, I told you short lecture. You have a time to go work on your term project.